All right. This is Dr. Wagoner. I hope everybody is safe and coping as best you can with our class being scattered by the coronavirus. And I know this is not the same as having me there in person. And trust me, I miss you too, sort of. And we will just try to get through this as best we can. And I'm still working on how to add video and things like that and try to make this a little bit more present. But as it is, here we are. And I'm going to be giving these lectures and I'm going to be breaking them up into smaller pieces because probably not everybody has really good internet connections. So I'm going to try to make these fairly simple. So you might recall that on Thursday, our last class meeting in person, we talked about transcription and translation, the way that a cell can take information that's stored in DNA and read that information and use it to assemble amino acids into a very well-defined sequence, the primary structure of a protein. And then that string of amino acids folds in a very complex ways that we don't fully understand, but that folds and takes up a shape. And once it has taken up its shape, it is able to do what it needs to do. And proteins do a ridiculous number of things in any living cell. So we've covered this. And I've compared this class to being kind of like climbing a mountain. Well, we're at the top of the mountain now, and there's lots of things we can look at. And we already talked on Thursday a little bit about viruses, but it seems very timely to make this the first thing we look at from the top of the mountain. A virus is not a cell. And it's an interesting philosophical question whether a virus can be said to be living or non-living. It's the kind of thing that you're supposed to debate in, philosophical BS discussions in your dorm room at 3 in the morning, if you still do that. A virus is a piece of DNA or RNA surrounded by a protein coat called a capsid. You can't see them with a light microscope. Maybe the very biggest might be visible, and sometimes they have funny consequences in cells that maybe you could see, but they're practically invisible. In isolation, they do absolutely nothing. You could take a jar full of viruses and it would not have any metabolism, it wouldn't take in oxygen, it wouldn't digest anything, it wouldn't excrete anything, it would sit there. But viruses inject their DNA or their RNA into host cells, which can be bacteria, protists, fungi, plants, animals, any life as far as I know. Even archaea can get them. And a cell that's been infected by a virus will start transcribing and translating the virus genes and making more virus DNA and making more virus proteins, and those self-assemble inside. And the cell becomes a virus factory, and the viruses eventually burst out of the cell, typically killing it, and spread to find other cells. And they can spread through a population of cells pretty rapidly. Some of them can. Virus capsids many of them are beautifully symmetrical, and they're made of proteins that are very tightly packed together. I picked this one because I thought it was pretty. It looks kind of like a psychedelic soccer ball. It happens to be the Zika virus. This is the one that was in the news a couple of years ago because it was transmitted by mosquitoes, showed up in the Caribbean, possibly in the U.S., and it potentially caused birth defects in the fetuses of infected women. So this was a real public health concern. but I just picked this because it happens to have a very attractive capsid, very regular. And here is a coronavirus. I don't think this is the COVID-19, which is the one we're all worried about now. It's a coronavirus. There are lots. Some of them only cause common colds, although the COVID-19 is a lot more serious. Its capsids happen to have these projections sticking out that you can see, looking a little bit like a crown, which is corona in Latin. And below, you can see a computer graphic reconstruction of what a complete virus particle would look like. Those prongs on the crown, those sticky outy things in the previous slide, are called fusion proteins. And what they do is they can clamp onto proteins that are expressed on the surface of a host cell. Cells have lots of proteins that are on the outside or sometimes bridging the outside and the inside of the cell. They do lots of stuff. They are a major way that cells can communicate with each other and respond to the environment that they're in. But viruses can take advantage of them to recognize host cells that they can infect and get pulled into the cell that way. So that thing that looks like colorful spaghetti from hell is coronavirus fusion protein that enables the virus to recognize, hey, there's a host cell right here, and the virus gets pulled into the cell and then begins taking the cell over. A virus can have DNA or RNA in it. Coronavirus is an RNA virus, not a DNA virus. And there's a special type of RNA virus called a retrovirus, so called because it wears bell-bottom jeans and likes lava lamps and the music of the Bee Gees. Actually, I'm kidding. That was a joke. Yeah, I can hear you laughing about as well as I could when we were in the lecture hall. No, a retrovirus includes not just RNA, but an enzyme called reverse transcriptase. And reverse transcriptase makes a complementary DNA strand from the RNA. That new DNA gets spliced into the host DNA, and then it gets transcribed and translated to make new retroviruses within the host cell. Now, what we call the central dogma of molecular biology is the idea that information flows in only one direction. It's stored in DNA. It is copied onto a piece of RNA by the process of transcription, and then it's used to make proteins by the process of translation. And it's a one-way process, except for retroviruses. Reverse transcription is the only time where you have information on a piece of RNA that's copied onto a piece of DNA. The far and away most famous example of retroviruses is HIV. And there is an electron micrograph of some HIV. HIV is being produced inside a white blood cell, one of the cells of your immune system, and budding off, and it wraps itself in cell membranes. Those little blobs that you see that are called budding particles and mature forms, those are HIV viruses that are wrapping themselves in a piece of membrane from the host cell as they go bursting out. Some viruses do that.